Saksham Narang is the co-founder and vice president of Yuva Biosciences and is part of the wave pushing age reversal technologies into the spotlight. So welcome, Saksham.
Thank you, and thank you for having us. It's nice to see a lot of familiar faces in the room, and for those that I don't know yet, I look forward to meeting you all in the networking event after this. But we're Yuva Biosciences, and we're based in Birmingham, Alabama, the real longevity hub. And we are really focused on mitochondrial science and breakthroughs in the field. And this really comes from someone that you all likely know, my second co-founder, Dr. Keshav Singh. Keshav has been studying mitochondrial science for nearly 3 decades now, and he calls himself a mitochondriac, and I guess all of us mitochondriacs as well. In fact, when I first met Keshav, I had just come back from Boston and moved back to Birmingham, Alabama, and Keshav had just put out a paper in Nature magazine detailing his work. I didn't know what quite to expect, so I waited outside the office to meet him to learn more. And sure enough, an SUV pulled up with a license plate that read Mito. Keshav stepped out in a mitochondrial tie and a mitochondrial pin. And I knew right there and then I was going to learn a lot about mitochondrial science and still continue to do that today. But in that paper that Keshav had published, he had developed a unique animal model in which he could regulate mitochondrial function system-wide or tissue-specific. So in essence, he could deplete mitochondrial activity system-wide in an animal and make them age. And then he could replete it. When he first depleted it, what he noticed was hair loss, skin wrinkles, inflammation everywhere. The female mice actually had abnormal estrous cycles, abnormal AMH hormone levels, and vaginal atrophy. Again, when he repleted it, all of these went away. So it set the stage for a very powerful cause and effect model that Yuva Bio built an entire platform on. And of course, you all are very familiar with this slide. In our highly unbiased opinion, mitochondrial dysfunction is a central driver of aging, and through the interactions with other hallmarks, it accelerates the deterioration of cellular health. So we focus on mitochondrial dysfunction, and we do that by measuring a range of biomarkers that address the root cause. We then, of course, look to validate that across collateral biological measures like oxidative stress response, inflammation modulation, and more. And I'll come back to that in a moment. But as I mentioned, one of the first things that Keshav noted when he depleted mitochondrial activity system-wide was hair loss and skin wrinkles. So we decided to focus on that market of topical treatments in beauty and cosmetics. And for 2 more reasons, right? Because there's probably no other industry that spends billions of dollars in promoting longevity and anti-aging other than beauty and cosmetic brands. Their bottom line depends on it. Second, everything that we see aging at the cellular level manifests onto the skin itself over time, right? We may feel something, then we see it, or we may see ourselves aging, then we begin to feel it. And specifically around mitochondrial dysfunction, there's several cellular consequences that occur that does have clinical implications for skin, for specifically like skin inflammation, hair loss, hair thinning, hair graying, skin wrinkles, and also aging spots, because mitochondrial dysfunction creates melanosome inactivity. So you see hair graying, and again, those aging spots. So we decided to focus on addressing the root cause of aging by identifying compounds and developing them that improve mitochondrial health and function. And after years of research, we did that with Yuva Bio Y100. Again, it's a natural compound because we're focused on topical treatments for the beauty and cosmetic sector. We discovered this, tested it several times in vitro, measured our biomarkers, and then we went and did more than 5 clinical trials with Yuva Bio Y100. In our hair aging study, we took 35 participants that were all clinically diagnosed with androgenetic alopecia, and we had them apply Yuva Bio Y100 topically twice per day as a leave-on product. Well, in this study, 82% of the participants had an increase in hair density. The average increase in hair density was about 30%. There was also statistically significant results for hair shedding and also in the improvement in the number of gray hairs. So these are 3 signs of hair aging that Y100, when applied topically, effectively addresses. We took the same Y100 and we formulated it into a skin cream and did a skin aging study. And in this one, we actually introduced a placebo control. And we did this through a split face study design where the participants applied Y100 topically to one side of the face, and then of course the placebo cream to the other side. And Y100 beat the placebo control across all 6 parameters of skin aging. Everything from wrinkles, elasticity, firmness, texture, evenness, and brightness as well. Again, this is the same Y100 now being applied across 2 aging trials for hair and skin. So we thought we've done it. We've got this clinical data. Let's go talk to brands. Let's go tell them about this. Let's showcase how we address the root cause of aging. And we did that. And we talked to a lot of brands, and we showed them our research. We showed them our science. We walked them through our mechanism. And then at the end of it, they said, so it's a super antioxidant. And we said, well, no, not quite. It addresses the root cause, but yes, excess free radicals is a byproduct of mitochondrial dysfunction. And so that's great. So it's a super antioxidant. Just go show us the antioxidant properties of this. And you all thought the FDA was difficult. That's a joke. But we did go back, and we took the Y100 we had now clinically validated, had our in vitro data on, and we went and measured its ability to scavenge free radicals. And Y100 actually beat the leading antioxidant in the market, and that it's 4 times more efficacious at scavenging free radicals. We went back, we shared this data, we started talking more to this brand. The next brand, he said, well, what about skin inflammation? You said that by addressing mitochondrial dysfunction, you should see an improvement in that. Where's the data to back that? So we went back, we tested Y100 and measured its response against pro-inflammatory cytokines. And Y100 was efficacious in addressing this by measuring IL-6 and IL-8. So we did that. We went, talked to another brand. They said, okay, well, great, what about collagen production? So we took Y100 that we've already validated now across these clinical studies, across all these different secondary measures, and we tested it for collagen. And Y100 also improved the CLIA1A1 gene that's responsible for collagen. So case by case, we continue to showcase that by addressing the root cause of aging, we're able to also have these secondary benefits as well that the industry is more likely to be familiar with. And then we started to announce our partnerships. And our first one is with Bosley MD, the leading hair restoration across the nation. They looked at Y100, they looked at our clinical data, and we worked with them to bring Y100 to market as a topical aerosol foam that's now available in market across these various channels that you see here. After launch, in just a few months, it quickly became a bestseller. The relationship with Bosley continues to grow, and they're excited about it and now have done a product expansion with us. So we have a topical serum that's just entered the market that's now exclusively available in Target. We have a lot more brand partnerships that we're looking to announce and product launches that are upcoming. And of course, we're not a one-compound company. We're also not looking at just one indication or 2 indications of aging. We're looking across the board, and that includes wound healing, that includes hair graying, of course, expanding on that data point. And we're also looking at women's health and more. We're also developing Y100 as a nutraceutical as well to look at intrinsic aging. So we have an entire platform, and really all of this is powered by what we call MitoNova. Through all of those repeat testings and measurements at both the gene and the protein expression levels at multiple time points in replicates, after 6 years of research, we had created enough data that we could finally train some models to have some predictive value in helping us identify the next series of Y100, Y101, Y102, and more. And that's what we continue to do today. We've added to our AI platform to add a couple of other development tools that we employ internally. And we actually have decided to use it in a high-throughput screening campaign where we told MitoNova, you can pick which compounds you want us to go and biologically validate. And it did this. So it screened a library of 700,000 plus compounds. It determined which ones were the ones that were most likely to be promoters of mitochondrial biogenesis. And we went and did a primary screen as well as a secondary screen to confirm at least 3 lead candidates that we're going to be moving into a preclinical study shortly. And that's actually taking Keshov's mouse model in that he's actually advanced that to be more tissue specific, and now we can actually reduce the activity of mitochondrial function and health in just the ovaries and study the cause and effect of that relationship. So we'll be taking one of these lead candidates and moving into that animal study shortly. We, of course, then realized that this tool could be more powerful if we allowed partners to use it and leverage it. And so we've announced this partnership with Yuva Biosciences, and they'll actually be using MitoNova to help them identify a compound for use in cardiometabolic conditions, including obesity. So I'll stop there in case anyone has any questions.
Questions? Irina?
Oh yeah, nice song. So the root cause of aging is not reactive oxygen species. It is mutations in mitochondrial DNA, which is 50% of mitochondrial proteins, right?
Mm-hmm.
So my prediction is that none of your compounds reverse mutations in mitochondrial DNA. Is it correct or not?
I mean, I think it's safe to say that we're not reversing mutations in mitochondrial DNA. But I will say one of the measurements that we look for and our biomarkers indirectly do, and we've also validated directly, is we see more mitochondrial DNA content when we do apply our compounds. So it may not be fixing or adjusting an existing mutation, but creating more healthy mitochondria could be just as an effect.
You cannot, right? So just FYI, you cannot create healthy mitochondria if mitochondrial DNA is mutated. It has been proven over and over again in developmental biology there are mitochondrial diseases and in aging. And I will stop here. We can discuss later.
We can discuss more and clarify that. Thank you.
With the hair product, do you have to cycle it at all and get it in phase with the anagen or telogen or exogen phase or any of that?
So we've done it was a 90-day study. Up until about 45 days, there was no improvement. But actually, what we did was we saw the efficacy at day 90. We haven't followed the participants in that study or followed up with them thereafter. But I will say that actually, by day 45, there was the number of people that actually were losing hair. So some of the ones that didn't experience a benefit, that rate actually diminished by day 90. So really, there was only one participant in that study that didn't have an improvement.
You mentioned that your formula uses all natural ingredients. I'd be curious if you could share what those are and if you've seen any studies about cultures that maybe naturally use those ingredients traditionally, and if they have experienced those benefits.
Yeah, well, it's a good point because a lot of this does come from traditional Chinese medicine around looking at natural products and botanical extracts. We initially had gone down that route in 2019, but we learned that working with extracts is just not, it's not a really great way to develop products. So we ended up, after going through a bit of screening and discovery, we did a little bit of fractionation, looked at the constituents, and then focused on the ones that we thought were the most bioactive, and then took it from there.